Welcome to Chapter 9, Muscles and Muscle Tissue, Part 2 of 3. In Part 1, we reviewed muscle tissue, the characteristics and functions of all types of muscle tissue, and then we shifted our focus to skeletal muscle. We studied the gross anatomy as well as the microanatomy. In Part 2 now, we are going to study the sliding filament theory, or we're basically going to see how muscle contracts. From there, we will bring in a very tiny portion of the nervous system. We will identify the neuromuscular junction and its parts, as well as the events that occur there that lead to muscle contraction. For the next few slides, we'll focus on the sliding filament model or theory of contraction. Remember, contractility was a characteristic of all muscle tissue. It is the ability of muscle tissue to shorten in order to generate force. But now we will look at contraction as activating cross bridges to generate that force at the microscopic level. We will bring in actin and myosin again, which you saw in Part 1. And when actin and myosin link up, they form a cross bridge. From that cross bridge, we see muscle contraction, and then we see that contraction end when that cross bridge detaches, or we pull actin and myosin away from each other. In Chapter 9, Part 1, we introduced the sarcomere. Remember that the sarcomere included a whole A band, which was mostly myosin or thick filaments, and half of an I band at each end. The majority of that I band was thin filaments or actin. In a relaxed state, so in a resting state, we really only see actin and myosin overlap briefly on the outer ends of the A band. But what happens during muscle contraction in response to stimulation from the nervous system, we form those cross bridges, or we link that myosin head up to the actin, and then they start to pull. The myosin will pull the actin towards the M line. And as that myosin pulls the actin towards the M line, we see those thin filaments slide past or over the thick ones, which is where we get the name sliding filament model. When thinking of the sliding filament model, you should think of a centipede's legs rather than a rowing team. These myosin heads reach up and grab the actin filament and pull them towards the M line, and reach up at another section and pull them again. A centipede's legs alternate each other, and that's how the myosin interacts with the actin and pulls on it. But what you truly need to know from this slide is the 5 bullet points at the bottom. This is what happens to the components of a sarcomere and of a myofibril during muscle contraction. Again, these filaments are not changing length. They are simply sliding across each other. So first, we have the Z discs that are going to be pulled towards the M line, or the middle. Therefore, the I bands are going to shorten because the actin is creeping in to the A band, which makes the I band shorter. The Z discs become closer to each other because the muscle is shortening. The H zone disappears. The H zone is the H zone because there is no actin there. But we will see on the next slide, during muscle contraction, the actin creeps into the H zone, making it disappear. And then lastly, the A bands move closer to each other. They do not change in length. On this slide, you should focus more on the cartoon rather than the microscopic image. Remember, in orange, you have the myosin or the thick filament, and in yellow and blue, you have the actin filament. But what you need to note here is what happens to the sarcomere during muscle contraction, which is seen in image 2. But in image 1, you have the H zone present, which is characterized by no actin. But as the myosin heads reach up or reach down, attach to that actin filament and pull it towards the M line to shorten the muscle, that actin creeps into the H zone, making it disappear. Again, the I band shortens, the Z discs move closer to each other, the H zone disappears, and the A bands move closer to each other but do not change length. Before any part of the sliding filament model can occur, and even before we can form a cross bridge, we need to talk about nerve stimulation, which goes back to the excitability or responsiveness characteristic of muscle tissue, as well as the neuromuscular junction. Before we see any contraction, we have the 4 steps that have to occur. The first is a nerve stimulus, so this comes from the nervous system. From there, we have the generation of electrical current, which we will now refer to as the action potential. The action potential is going to be created in the sarcolemma, or the plasma membrane of a muscle cell. From there, we need to spread that electrical current all across the entire sarcolemma, and from there we see a rise in the intracellular levels of calcium. You can break these 4 steps up into 1 and 2, and 3 and 4. 1 and 2 we will see occur at what is called the neuromuscular junction. 3 and 4 will be occurring within the muscle fiber. And 3 and 4 are important because they link that electrical current to the actual shortening of muscle. And we will see that 3 and 4 are referred to as excitation, contraction, coupling. While we don't cover the nervous system in EXS216, we do mention it briefly because that is the system that we use to stimulate muscle contraction. So skeletal muscles like the quadriceps or the hamstrings are stimulated by these structures, or power lines rather, known as somatic motor neurons. Remember, neurons are the primary cell type of nervous tissue. They are motor because they control muscle. And the term somatic means body. These muscles move our bodies. Those neurons are made up of axons. So if you think about a box of spaghetti, you reach your hand in and you take out 12 noodles. 12 of those noodles together make up 1 neuron, but each noodle is an axon. Those axons serve as the power lines for that electrical current to go from the nervous system to skeletal muscle. Each of those noodles, or each of those axons, will branch as it enters the muscle. And as it branches, it's forming the neuromuscular junction, or the communication point between a nervous structure and the muscle. As always, here's a picture of the neuromuscular junction if you are having difficulty trying to figure out what it actually is. So at the very top of the screen, we have an axon. The axon makes up a neuron, which is a part of the nervous system. The axon will branch, forming axon terminals, and here we can see 3 of them. They are all going to communicate with or synapse with a muscle fiber, or the sarcolemma of a muscle fiber. Now, if we take one of those communication points or one of those synapses, that is what we are looking at in the bigger picture. This yellow bouton structure is the axon terminal. The axon terminal will communicate with the sarcolemma of a muscle fiber. And this is where we see that electrical event occur prior to muscle contraction. Overall, the neuromuscular junction is that point of information transmission or communication between the nervous system and a muscle or the muscular system. There are a few components to the NMJ or to the neuromuscular junction, and the first is the axon terminal. Remember, each axon of a neuron will divide or branch into many axon terminals. Now, that axon terminal is not directly stuck to the sarcolemma. There is a little space in between the two structures, known as the synaptic cleft. In each of those axon terminals before the cleft, we find synaptic vesicles, little bubbles that contain a neurotransmitter known as acetylcholine. You will see it listed as ACH from here on out. Now, moving across that synaptic cleft or that space, and now looking at the sarcolemma or the plasma membrane of a muscle fiber folds in on itself. It folds up and then it dives down. These are called junctional folds, and the purpose of them is to increase the surface area, but also to have a location for these receptors that will bind acetylcholine once it is released from the vesicles in the axon terminal. So overall, the neuromuscular junction will include axon terminals, the space known as the synaptic cleft, as well as the junctional folds on the sarcolemma. Now, taking the information that I just covered on the previous slide, we are now able to identify all of the parts and pieces of the neuromuscular junction. So if you are following along with my laser, again, in the yellow, that is an axon terminal. In the axon terminal, we find bubbles or vesicles. These vesicles contain a neurotransmitter known as acetylcholine. As you move away from the axon terminal, you now find a space or a gel-filled space known as the synaptic cleft. And as you move through that space now, you find the junctional folds on the sarcolemma of a muscle fiber. Within those folds, we find receptors where acetylcholine will bind to. Now, when we put all of the parts and pieces together at the neuromuscular junction, we can see how we have nerve stimulation and the generation of an action potential and sending that signal to the muscle. So first, this nerve impulse or this electrical current reaches the axon terminal. When that electrical current reaches the terminal, it stimulates the release of acetylcholine. So think of these vesicles popping and releasing that important neurotransmitter into the cleft or into the space. Acetylcholine will move across the cleft and find the acetylcholine receptors within the junctional folds on the sarcolemma. Once acetylcholine binds to its receptors, we will see that electrical event continue across the sarcolemma, and we will see the generation of an action potential throughout the entire muscle fiber. Now, it's important that we get rid of some of this acetylcholine, otherwise we would have constant muscle contraction. So this is where we see an enzyme known as acetylcholine esterase come into play. It is going to break down any excess acetylcholine to make sure that the muscle contraction stops. Now you might be wondering, how do we have electricity in our body? And if you remember from Unit 1, we talked about the membrane potential, or this term Depolarization. And this comes about because there's a voltage difference at the plasma membrane or across the plasma membrane of a cell. If you were to stick a voltmeter inside of a cell, it would measure about -50 to -90 millivolts. And that means that the inside of that cell is more negative compared to the outside. This also occurs when a cell is at rest. And when a cell is resting, it is said to be polarized. This term action potential now, or electrical current, is brought about due to changes in electrical charges. And changes in electrical charges are caused by the openings of channels, which allow for the flow to ions, or allow for the flow of ions. We will see 3 steps in order for us to generate this electrical current, which we will now call an action potential. We have the end plate potential, depolarization, and repolarization. In order for us to generate an electrical current or an action potential, we first need to have an end plate potential. So going back to the events at the neuromuscular junction, we have acetylcholine released from the axon terminal, which will diffuse across the cleft to bind to acetylcholine receptors on the muscle fiber. Once acetylcholine binds to the muscle fiber, that is a chemical messenger that will cause the opening of chemically gated channels. Once we open these channels, we have sodium come into the muscle fiber. And as sodium flows into the muscle fiber, it brings with it its positive charge. And as more positive charge comes in, we are driving that overall value of the inside of the cell towards 0 or towards threshold. We are stimulating it. Again, to reiterate, we have the axon terminal in yellow. Within the axon terminal, we have synaptic vesicles, which contain acetylcholine. Acetylcholine will be released from the axon terminal. It will diffuse across the cleft. Once it diffuses, it will bind to acetylcholine receptors on the sarcolemma. Acetylcholine is stimulatory. It is going to stimulate the opening of chemically gated ion channels. These channels are now going to open and will allow sodium to enter the cell. As sodium enters the cell, it brings with it its positive charge, making the inside value of the cell go from -70 to maybe -50, making it less negative, driving it towards threshold, or exciting it. Now that we have our end plate potential, and you should think that the end plate potential is very localized, but when it comes to sodium, we see a positive feedback loop. So with that end plate potential, we see sodium coming into the cell, and as more comes in, these channels that are adjacent begin to open as well. All of this sodium coming in brings with it its positive charge, so it may take the inside value of the cell from -70 to -50 millivolts, driving it towards threshold. Once we hit threshold, that triggers an action potential, and we will see the propagation or the spreading out of this action potential across the entire sarcolemma of the muscle fiber. Here's an image to hopefully help you put into perspective what depolarization is. When we depolarize something, we are continuing to generate that electrical current, but also spreading it across the entire cell. So on the left, we've seen our end plate potential. Once acetylcholine binds to its receptors on the sarcolemma, it stimulates the opening of sodium-gated channels. Once these channels open, sodium enters the cell, bringing with it its positive charge. Here is a negative feedback loop. As sodium is coming into the cell, it stimulates the opening of more sodium channels, meaning more sodium comes in, driving that value inside of the cell closer to threshold so we can continue to generate as well as spread out that electrical current. Now, the last bit of this excitation portion of this excitation contraction coupling comes from repolarization. When you hear the term repolarization, you should think returning back to baseline. In order to bring that inside value of the cell back down or more negative to where it once was, we are going to close our sodium channels and open our potassium channels. When potassium channels are open, potassium wants to leave the cell. And when it leaves the cell, it takes with it its positive charge, making the inside of the cell more negative. As the inside of the cell becomes more negative, keep in mind we are moving away from threshold. Threshold is how we generate action potentials. So as we move away from threshold, this is known as the refractory period, where we cannot stimulate that muscle fiber. There are no sodium channels available to be open, so there's no more generation or even propagation of another action potential. As we have these ions moving about, we have to bring into play the sodium-potassium pump. The sodium-potassium pump will restore ionic conditions by putting sodium and potassium where they need to be, not where they want to be. On this slide, we'll tie everything together. So in the image that you've seen before, number 1, this is an end plate potential. It's a localized stimulation. It comes from the release of acetylcholine from the axon terminal. Acetylcholine diffuses across the cleft and binds to the sarcolemma. As it binds to the sarcolemma, it stimulates the opening of sodium-gated channels in the adjacent regions. Now we move to step 2, depolarization. As sodium enters the cell, we see positive feedback. Once sodium enters the cell, we see more sodium come in and more channels open, which drives the inside value of the cell towards threshold so we can continue to generate as well as propagate that action potential or that nerve impulse. Now, once all of our sodium channels are open, we begin to see step 3, repolarization. We are going to return to baseline. So we close our sodium channels and we open our potassium channels. When we open our potassium channels now, potassium leaves the cell, taking with it its positive charge, bringing the inside of that cell back down away from zero, away from threshold, where we also see the refractory period, where it's rather difficult, sometimes impossible, to generate another action potential. Now, if you're having trouble putting these words and these diagrams together, this graph shows you the voltage inside of the cell in relation to the end plate potential, depolarization, and repolarization. So if you start on the left side of the screen, we see that our muscle cell is polarized or is resting at negative 90 millivolts. We have a nerve stimulus. So we have acetylcholine released from an axon terminal. It diffuses across the cleft and binds to the sarcolemma. As acetylcholine binds to the receptors, it stimulates the opening of sodium channels. Now, take a look at where the graph begins to creep up. As the graph begins to creep up, sodium channels are opening. Sodium is entering the cell, driving that inside value towards zero or towards threshold. Eventually, we see depolarization. We have reached threshold. We have enough energy, enough stimulus to spread and propagate that nerve stimulus. When you reach the peak, all of our sodium channels are open, so we begin to close them. And as we close our sodium channels, we open potassium channels. Potassium leaves the cell, driving that value away from 0. This is now repolarization. We are returning back to baseline. This was Chapter 9, Part 2. Again, we introduced the sliding filament model. We briefly discussed a nerve stimulus and the nervous system. We talked about the bits and pieces of the neuromuscular junction and the events that occur there. And then we finished up with the generation and propagation of the action potential along the sarcolemma.